Welcome back. I want you to think back to February 2014.
Oh yeah, a big year.
Right. If you were following the fast food industry or just, paying attention to how our food culture was shifting, you might remember Chick-fil-A making this pretty massive promise.
The antibiotic-free pledge.
Exactly. They announced that by 2019, they were going to transition their entire supply chain to serve only chicken raised without antibiotics.
Which was huge.
Right. I mean, places like Panera or Chipotle had already made similar pledges, but Chick-fil-A doing it, that moved the needle. It signaled this unavoidable shift for the whole agricultural industry.
Yeah, and at the time, it was marketed heavily as this pivot to, natural food.
Lean eating, yeah.
But behind the PR campaigns, it really wasn't just a trendy marketing thing. It was a direct response to some incredibly dire warnings.
From the global public health folks.
Exactly. It was an epidemiological reality basically catching up with the supply chain. Throughout the 2010s, the World Health Organization and the CDC were aggressively sounding the alarm. They were tracking this terrifying acceleration of antimicrobial resistance. Basically, bacteria that were completely unfazed by the antibiotics we use to eradicate them.
And I think the stakes here, they're almost impossible to overstate. For the last 80 years, we've had this luxury of antibiotics.
A total luxury.
They are the foundational medical miracles, really. They make modern surgery, chemotherapy, basic infection control possible. But this rising tide of resistance threatens to drag humanity backward.
Back to a pre-penicillin era.
Right. An era where a routine surgery, or honestly, even a simple paper cut, could become a lethal, untreatable threat. Okay, let's unpack this.
The mission for this deep dive is to synthesize the architecture of this entire crisis. Look at the dual forces here.
Human misuse and agricultural overuse.
Right. How those two things drove antimicrobial resistance to a breaking point. But we're also going to explore the cutting-edge solutions.
The stuff that doesn't just rely on making more of the exact same failing drugs.
Exactly. The scientific and economic paradigm shifts.
So consider this your comprehensive shortcut to understanding a massive global vulnerability. We're going to cover some bizarre historical missteps.
Some very weird history.
Some brilliant aha moments in science. And we'll structure it all to respect your time. So to really grasp the gravity of antimicrobial resistance, or AMR, we have to start small.
Very small, the microbiological level.
Right. It boils down to selective pressure. We flood an environment with antimicrobials, and that successfully kills off the susceptible pathogens.
But the survivors, the germs possessing a specific genetic resistance mechanism, they survive.
And they multiply.
Right. And they pass those specialized traits on. We are essentially engineering our own vulnerability.
Wow.
By deploying antibiotics at this massive industrial scale, we are inadvertently selecting for the most resilient traits. We ensure that the bacteria with the most robust survival mechanisms are the only ones left to dominate.
And when you look at the defense mechanisms these bacteria have actually developed, I mean, the biological warfare happening at a microscopic level is wild.
It's highly sophisticated.
There are 4 primary strategies they use. The first one is altering permeability.
Right, because to be effective, an antibiotic molecule has to physically enter the bacterial cell.
Makes sense.
Gram-negative bacteria, for example, have an outer membrane equipped with these things called porins. They act as cellular gateways.
And I love this analogy from our research. There's a brilliant way to visualize those porins. Picture them looking exactly like those green warp pipes from Mario.
Yes, the Mario pipes.
Right. So when faced with a threat, like, say, carbapenem antibiotics, the bacteria essentially seal off the pipes. Or they change the shape of the pipe so the drug can't fit down into the cell.
They downregulate their porin expression, severe restriction of the drug's concentration inside the cell.
But what happens if the antibiotic molecule actually manages to get inside? Like it slips past the pipe?
That brings us to the second mechanism, efflux pumps.
Efflux pumps.
Right. These are specialized transport proteins in the cell membrane. The bacteria use them to actively expel toxic substances.
They just spit it out.
They literally pump the antibiotic back out into the extracellular space before it can reach its target. And what makes these efflux pumps so insidious is that a lot of them are multi-drug transporters.
So they don't just work on one drug.
Exactly. A single pump can expel multiple structurally unrelated classes of antibiotics.
That's terrifying. And then there's the third defense, which is all about modifying the target site itself.
The lock and key mechanism.
Right. So if we look at penicillin, it works by binding to a specific enzyme called transpeptidase. The bacteria need that enzyme to cross-link their cell walls.
It's the lock, and penicillin is the key.
Exactly. But the bacteria counter this by mutating their genetic sequence. They alter the lock. They change the enzyme structure just enough that the penicillin key no longer fits.
The drug is rendered completely useless.
And if modifying the target isn't an option, they deploy the fourth strategy,
Enzymatic degradation.
basically breaking the key.
Yes. They synthesize their own enzymes to actively seek out and destroy the antibiotic molecule. They dismantle its chemical structure.
It's like penicillinase.
A classic example. Penicillinase specifically breaks open the beta-lactam ring of the penicillin molecule, deactivating it entirely.
But the thing that really gets me, the truly terrifying part, is that bacteria didn't just invent this weapon from scratch the moment we introduced modern medicine.
What's fascinating here is that penicillinase existed in nature long before human beings ever discovered penicillin.
It was already out there.
It's an ancient evolutionary mechanism. We didn't induce the creation of this enzyme. We merely introduced massive, unprecedented volumes of the antibiotic into the global ecosystem.
And that exponentially accelerated the dominance of the bacteria that already had the trait.
Precisely. We selected for it.
And these traits, they spread with terrifying efficiency. They don't just pass them down from parent to offspring. They engage in horizontal gene transfer.
Often utilizing plasmids.
Plasmids.
Yeah.
Now, there's a perfect analogy for how plasmids spread through a bacterial population.
The CD analogy.
It is exactly like the early 2000s when you'd burn a CD. Imagine you burn a My Chemical Romance album onto a CD and hand it to a friend.
Classic.
Right. They rip the tracks to their computer, and then they burn a fresh copy for someone else. Plasmids work the exact same way.
Because plasmids are extra chromosomal genetic elements, they replicate independently of the host's chromosomal DNA.
They're just passing the mixed CD around.
And that CD can carry a single resistance gene or multiple genes at once, a highly efficient mixed CD of multi-drug resistance.
And they can even cross species barriers, right?
Yes. Entirely different groups of bacteria can share these resistance traits. And it gets highly optimized when bacteria form biofilms.
Biofilms are basically clumps of cells.
Right, structured consortiums of cells sticking to a surface, protected by a self-produced matrix. Inside that dense protected community, plasmid transfer happens at a dramatically accelerated rate.
Which brings us to the human element. We essentially place bacterial evolution on a high-speed medical treadmill.
We sped the whole thing up.
You'd think widespread resistance took decades to show up, but the timeline is shockingly compressed.
Very compressed.
Before penicillin was even universally available, scientists isolated a resistant strain of strep in a military hospital in the late 1930s.
And by the 1940s, researchers documented a strain of E. coli producing penicillinase.
And that was before they even had definitive clinical evidence that penicillin would work in humans.
The pioneers of the era were acutely aware of the danger. Alexander Fleming himself.
The guy who discovered the penicillium mold.
Yes. In his 1945 Nobel Prize lecture, he issued a stark prophetic warning. He explicitly cautioned against the quote-unquote ignorant man who might underdose himself.
He knew exactly what would happen.
He understood that exposing microbes to sublethal concentrations wouldn't kill the infection, but it would provide the exact selective pressure needed to forge resistant strains.
But the cycle was already in motion. Pharma rolls out a novel drug, doctors prescribe it, selective pressure builds resistance, and then everyone scrambles for a replacement.
It was a constant race. Tetracyclines hit the market in the early 50s.
And by 1959, we had resistant Shigella.
Vancomycin was introduced in 1958 as a drug of last resort. It held the line for nearly 30 years, but eventually enterococci evolved to beat it.
To keep up, researchers had to look in some pretty bizarre places. This is the story of Giuseppe Brazzu in 1945.
The Italian academic.
Right. He was working in Sardinia, and he hypothesized that to find new antibiotics, you need highly competitive environments.
So he went to the raw sewage outflow.
He started sampling raw sewage. The incredible insight here is that humanity literally relied on wastewater to outpace bacterial evolution.
And it worked?
In that Sardinian sewage, Brazzu isolated a microbe that produced cephalosporin.
A novel class of antibiotics, highly effective against penicillinase-producing bacteria.
We bought ourselves a few more decades using literal waste. But bioprospecting in nature wasn't fast enough.
No, the industry had to shift towards synthetic biology, tweaking the molecular structures of existing drugs to bypass resistance.
So in 1959, chemists synthesized methicillin.
A beta-lactam antibiotic designed specifically to withstand penicillinase.
And if you've ever had to scrub down with that harsh pink antiseptic soap before hospital surgery, this next part is exactly why.
The victory was short-lived.
Incredibly short-lived. Just 2 years later, in 1961, a London researcher named Patricia Jevons documented a staph strain that completely ignored methicillin.
The dawn of MRSA, methicillin-resistant Staphylococcus aureus.
The superbug.
The bacteria had acquired an entirely new resistance mechanism. They altered their penicillin-binding proteins so significantly that the new synthetic drug had zero affinity for the target.
So the strategy shifted to combination therapies, mixing drugs together,
Like in 1967 with Beecham Pharmaceuticals, they researched beta-lactamase inhibitors
which led to pairing amoxicillin with clavulanic acid.
to create Augmentin. It's a fascinating biochemical tag team. The clavulanic acid has minimal antibacterial activity on its own, but it acts as a suicide inhibitor.
It sacrifices itself.
Yes. It permanently binds to the bacteria's defensive enzymes, neutralizing them, so the amoxicillin can successfully destroy the cell wall.
Okay, here's where it gets really interesting. Because while human medicine was fighting this highly sophisticated war in hospitals, a massive, poorly regulated experiment was unfolding in the American agricultural sector.
The post-WWII poultry boom.
Domestic demand skyrocketed. The industry wanted massive yield. They even had a national breeding competition called the Chicken of Tomorrow.
They needed meaty birds, and they needed cheap vitamins to scale up production, specifically vitamin B12.
Which leads us to 1948 and a nutrition researcher named Thomas Jukes. He was working at Ladurl Laboratories.
Ladurl had recently developed aureomycin, the first commercially viable tetracycline antibiotic.
And Jukes knew that the bacteria used to make the antibiotic also synthesized vitamin B12 as a byproduct.
So he ran a trial on malnourished chicks. He fed the experimental group the leftover aureomycin mash,
Literally the industrial waste product from manufacturing the antibiotic.
and the physiological results defied all expectations.
The chicks didn't just survive. They exhibited explosive physical growth. They outpaced the control group that was getting pure liver extract.
So Jukes started supplementing their feed with pure powdered aureomycin.
Unprecedented weight gains.
If we connect this to the bigger picture, Jukes' discovery birthed an entirely new, highly ambiguous regulatory category.
Growth promoters.
Right. These subtherapeutic doses weren't medicine for sick animals, and they weren't strictly nutritional. They were growth promoters.
And seeing the massive economic upside, the FDA formally approved antibiotic feed additives for growth promotion in 1951.
And Ladurl Laboratories pushed the envelope even further with a process called acronization.
This part is wild. They were quite literally dipping raw, processed poultry carcasses into tanks of diluted aureomycin solution.
Just to extend shelf life before shipping to grocery stores.
And the epidemiological fallout happened fast. In 1956, an epidemiologist named Reimert Ravenholt investigated an alarming surge of severe staph infections.
Among blue-collar workers in Seattle.
He did the contact tracing and found the only common denominator was their employment at a local poultry processing facility.
A facility that had just implemented the acronization process.
Ravenholt mapped out this terrifying new vector for disease. The low-dose antibiotic baths weren't sterilizing the meat.
They were applying massive selective pressure.
The wash eliminated all the susceptible bacteria, leaving behind an incredibly concentrated population of highly resistant superbugs.
And when the workers handled the acronized poultry, those superbugs colonized the minor abrasions on their hands.
It proved definitively that humans didn't even need to eat the antibiotics to suffer the consequences of farm-level superbugs.
Yet despite this clear evidence, the global regulatory response was deeply fractured.
To say the least.
In the late 50s, Japanese researchers identified R factors. We now know them as plasmids carrying multiple resistance genes. This proved resistance could spread rapidly without continuous exposure to the drug.
Then in 1969, the UK government published the Swan Report. It laid out incontrovertible evidence that routine agricultural antibiotics cause human resistance,
Leading the UK to formally ban agricultural growth promoters in 1971.
and the US tried to follow suit. In 1977, the FDA commissioner, Donald Kennedy, pushed for a comprehensive ban on farm use of vital human antibiotics.
But the proposal met intense, organized resistance.
A huge coalition of pharmaceutical manufacturers and agricultural conglomerates lobbied hard, and just to report impartially on their stance, their primary argument was that the epidemiological link between farm use and human mortality wasn't definitively proven.
They claimed the science wasn't settled.
And because of that gridlock, The ban completely stalled out. The U.S. government didn't take systemic action until 2013,
When the FDA issued guidance for Industry 213,
which requested a voluntary phase-out of growth promoters.
which perfectly contextualizes that 2014 Chick-fil-A announcement you brought up at the start.
Right. They were adapting their supply chain just as the FDA was finally closing the loophole.
But those decades of delay have left us in a highly precarious position today.
The numbers are grim. Over 1 million deaths globally each year directly implicated by AMR pathogens.
In the U.S. alone, roughly 35,000 annual fatalities.
And our pharmaceutical arsenal is severely depleted. We used to have around 155 FDA-approved antibacterial compounds in the pipeline. Today, that active pipeline has collapsed to roughly 90 to 100 compounds.
And the root cause of this empty pipeline isn't a lack of scientific capability. It is a fundamental failure of the economic model.
It's just a terrible business model.
Uniquely punishing. It requires an average capital investment of $1 billion.
$1 billion.
And roughly a decade of clinical development. With a staggering attrition rate. Nearly 90% of candidates fail when moving from preclinical to human trials.
Usually due to toxicity.
Or poor pharmacokinetic profiles.
And even if a company achieves the impossible spending 10 years and $1 billion to get a safe, effective antibiotic approved by the FDA doctors, actively try not to use it.
Antimicrobial stewardship programs mandate that physicians hold these new drugs in reserve.
As a last resort.
Exactly, to delay the onset of resistance. From a fiduciary standpoint, it's an untenable model for big pharma. Why invest $1 billion into a curative drug that patients take for 5 days and that hospitals intentionally refuse to buy in bulk?
Especially compared to a daily maintenance pill, like Lipitor for cholesterol. You take that continuously for decades.
Antibiotics offer a terrible return on investment. So the burden has shifted away from big pharma to academic institutions and boutique biotech firms.
So what does this all mean? How do we restructure the system to prevent a post-antibiotic era?
On the scientific front, we're seeing profound technological leaps. For decades, microbiologists were bottlenecked because most soil bacteria prime candidates for new antibiotics won't grow in a standard petri dish.
But in 2010, researchers developed the iChip.
The iChip is a brilliant piece of bioengineering. It's a microscopic grid isolating individual bacterial cells in their own tiny chambers,
And they placed the chip back into the native soil.
allowing the unculturable bacteria to grow using the natural nutrients of their own ecosystem.
Which unlocked a massive biological library. Using the iChip in 2015, a team isolated a new soil bacterium producing a compound called texobactin.
And its mechanism of action is entirely different from legacy drugs. It binds to highly conserved lipid precursors of the bacterial cell wall.
In clinical evaluations, it rapidly eradicated MRSA. And because its target is so fundamental to the structure, pathogens will struggle to develop resistance.
Alongside that, we're leveraging massive computational power, whole genome sequencing to map a pathogen's genetic code before a single drug is administered.
And artificial intelligence. AI models are predicting evolutionary mutations, identifying resistance patterns before they manifest in a clinic.
But all these breakthroughs will wither without a viable economic framework. Policy experts are developing push and pull incentives to bridge the gap.
Push incentives subsidize the massive R&D costs up front. Pull incentives guarantee a financial return if a drug reaches the market.
One of the most promising pull incentives is the FDA subscription model.
The Netflix model for antibiotics.
Right. Instead of paying per pill, government health systems pay a massive, flat annual subscription fee for access to the novel antibiotic.
Decoupling profit from sales volume. They recoup their billion-dollar investment even if the drug sits on a shelf in reserve.
There are also proposals for market entry rewards, government lump sum awards, where they essentially buy the patent outright upon approval.
And on the push side, we have bioaccelerators like CRRBX in Boston. They function like a Silicon Valley tech incubator, but for early-stage biotech firms focused on novel antimicrobials.
Ultimately, analyzing this crisis requires the One Health approach.
We can't treat human medicine, veterinary science, and environmental regulation as isolated things anymore.
The health of our agricultural supply chain is inextricably linked to the efficacy of human medical interventions. You cannot flood the farm with antibiotics and expect the pharmacy to remain secure.
It has been a wild evolution to get here. From Giuseppe Brazzu isolating cephalosporins in Sardinian sewage, to Mario Pipe cellular gateways, to the disaster of oreomycin-soaked poultry.
And all the way to AI drug hunters.
It's a testament to human innovation clashing with the relentless adaptability of bacterial evolution.
Which leaves us with one final, incredibly provocative concept to consider from our research today.
What's that?
We've spent this entire deep dive discussing the synthesis of chemical compounds to eradicate bacteria. But what if the ultimate solution to the AMR crisis isn't a synthetic drug at all?
Okay.
What if we pivot to a discipline heavily researched during the Soviet era and deploy bacteriophages?
Phages.
Naturally occurring, living viruses that have evolved specifically to hunt, infect, and destroy bacterial cells.
The ultimate biological counteroffensive.
A literal enemy of my enemies scenario. It replaces static chemical treatments with dynamic, living predators. It flips our entire pharmacological model on its head.
And it might just be the paradigm shift we need to win this arms race. A living virus deployed to hunt down multi-drug-resistant superbugs. Now that is something to think about. Until next time, keep questioning, keep learning, and we'll see you on the next Deep Dive.